So when Mendel was studying garden peas, he could just manipulate the parents. But of course, when we study human diseases and conditions, we can't just manipulate who mates with who. And so we rely instead on something called pedigree analysis. Pedigree analysis is kind of like a family tree. You create a chart that shows relationships between individuals and the offspring that they had over a couple of generations. But instead of following the names or, the way a family tree is, it's really focused on disease. So in a pedigree, we'll use symbols to represent whether somebody had a condition or not. So for example, we're going to use a circle or a square. The circle is for female, the square is for male. If the circle or square is open, then those individuals are unaffected by the disease or condition or trait that you're following. If they are affected, then they are filled in. So that means they have the disease or trait that's being followed in the tree. In some pedigree analysis, you'll see half-filled individuals. This means that they're a carrier, but this is not always marked. So in most pedigrees, you're not going to see carriers indicated because you're supposed to figure out who's a carrier based on the pedigree. So just keep that in mind, that you're usually not going to see carriers marked. So diseases are classified into 3 categories: autosomal recessive, autosomal dominant, and sex-linked. And so the first part of the name, autosomal, means that it's on regular chromosomes. So autosomes are chromosomes 1 through 22, basically not the sex chromosomes. The recessive part means that in order to have the condition or the trait, you have to be homozygous recessive, or you have to have the 2 alleles. So, sorry, my pen's not quite working here. Let me get this over here. So somebody with the condition, if it's recessive, has to have the 2 recessive alleles. Anybody who's unaffected has either the 2 dominant alleles or they're a heterozygote. So again, if a disease is autosomal recessive, anybody who's affected is homozygous for the trait. Anybody who is not affected can either be homozygous or heterozygous, because being normal, if it's a disease, is dominant. So when you look at a pedigree, you are sometimes not going to know whether the disease is autosomal recessive or not. And so when you look at a pedigree, you're going to be looking for certain trends in the pedigree to tell you whether or not it's recessive. And one of the key, some of the key things that you'll see in a pedigree if a disease is recessive is that it's relatively rare, and affected children can have unaffected parents. So that's one of the keys. It's also sometimes called skipping generations. So notice that this child has the condition, but both their parents do not have the condition. Same thing over here on the tree. These children have the condition, but neither of their parents have the condition. So it will appear in the children even if the parents do not have it. When that happens, that means that both parents must be carriers. So when somebody is a heterozygote for an autosomal recessive disease, they're called a carrier. That means they're carrying the disease gene, but you can't see it in them. So again, they don't have the disease, but they're carrying it and they can pass it on to their children. So if 2 carriers get together, that's how you can have children that get the disease. So the other thing we're going to do with pedigrees is fill in genotypes. So when you're filling in an autosomal recessive genotype, or the genotypes for an autosomal recessive disease, you want to start with the people who have the disease and work backwards from them. So if these people are normal, they must have at least 1 normal allele, but because they each gave a disease allele to their child, they must both be carriers. Same thing with this person. We know that they must be homozygous recessive. These parents are normal, but they must be carrying at least one disease allele because that's how their child got it, right? And then these two individuals we know must have at least one normal allele, but we can't really say about their second one, so we just put a question mark. Same thing over here. We know that they must have at least one normal one, but we can't really say about the second one. There's no way to know. So that's how you fill in the genotypes. Some diseases or conditions are autosomal dominant. So again, that means that it's not on a sex chromosome, so not carried on a sex chromosome, but the trait is dominant. If a trait is dominant, then affected individuals can be heterozygous or homozygous, and unaffected individuals are homozygous recessive. So this is the opposite of what we saw with an autosomal recessive disease. If you're looking at a pedigree, the pattern that you're going to tend to see within the pedigree, so within a family, not the whole population per se, but within a family, the trait will be more common. Rather than rare, as it was in an autosomal recessive disease. The other thing is that affected children have affected parents. So remember, with recessive diseases, it could skip generations. But if I look here, here's a child with the disease, here's the parent with the disease. Here's a child with the disease, here's a parent with the disease. Here's a child with the disease, here's a parent with the disease. Here's a child with the disease, here's a parent with the disease. Now notice that that's true when you work backwards, but just because somebody doesn't have just because a parent has the disease doesn't mean the kids do. So notice that neither of these kids have the disease, even though their parent does. So you can have children that don't get the disease, but if you have an affected child, there should be an affected parent. So that's kind of the key ways to tell that something is autosomal dominant. So you can also fill in genotypes. So in the case of an autosomal dominant disease, it makes sense to start with the people who don't have the condition because they must be homozygous recessive, absolutely. So you can go in and fill in anybody who does not have it. And then you can work backwards from them. So this person has a disease, so they must have at least one of the dominant alleles. But because their child does not have it, they got each one of these from one parent, so one of those must have come from this parent. Same thing here. This person has the disease allele, but because they have children that don't, they must be heterozygous. And then this person has the disease. They must be heterozygous because they have a normal parent, and that parent must have given them that. Same thing with this person. They got the disease allele, but they have a normal parent, so they must have given them one normal one. Same with this one, right? And then again, same with this one. So you can just logic it out that way, and we can figure out everybody in this family pedigree compared to the last one we looked at. So autosomal recessive and autosomal dominant conditions have to do with the disease being carried on, again, the autosomes, which means not the sex chromosomes. So sex chromosomes are going to change inheritance if the disease is carried on the sex chromosomes. So I just want to talk for a second about sex chromosomes, and then we'll look at how that affects a pedigree. So in humans, we have an XY system of sex determination. So the sex chromosomes determine the sex of the individual. Males are XY and females have two X chromosomes. This is not the only mechanism in nature. You see an XO system in birds, for example, a ZW system in birds rather, an XO system in insects. And then even some other systems. There's some reptiles where the sex is determined by the temperature of the nest. Bees have a really different system where the male is haploid and the female is diploid. So there's other systems for sex determination. We're going to focus on the XY system and how that affects pedigrees. So because men only get one X chromosome, they are hemizygous for any traits on the X chromosome. That means they only get one copy. For every other chromosome, everybody gets two copies, right? So they can be either heterozygous or homozygous. But for X chromosome traits, men are hemizygous. They only get one copy. So just want to remind you that the X chromosome and the Y chromosome are very different from each other. So they look very different. They don't carry the same information. They are not homologous. And basically, and I know I've said this before, all you need to be human is one X, right? So what that means is all important information for being human, like the key information for being human, is on the X chromosome and just on one of them, right? So you only need one X to be human. The Y chromosome mainly carries some male-specific traits. So things like sperm production, things that control fertility. But it's not going to lead to any diseases other than fertility problems. So as far as studying human conditions, we're primarily talking about the X chromosome. So traits that are carried on the X chromosome are called X-linked traits. And again, this is significant because men will only get one copy. So they can't be homozygous or heterozygous for X-linked traits. They're just going to have one. If you think about it, they only get one chance to get a good version of the gene or a functional version of the gene. So they are more likely to get X-linked diseases because, again, they only get one chance to get a normal copy. Women get a chance to have two chances to get the normal copy. So they're less likely to have a X-linked disease. So the key thing that you will see on a pedigree that will tell you that it's probably X-linked is that males will be much more affected. Usually, not always, but usually you're only going to see males with the condition. Examples of this are things like color blindness and hemophilia. These are relatively rare in females, but not that unusual in males. And again, that's because men can only get that one copy, so they have a much greater chance of getting a messed up version of it. You can do punnett squares with sex-linked traits, just like you do with regular traits. The only thing is that you're going to now track sex chromosomes at the same time. So if you have a normal male for a trait, so this is for hemophilia, so he has the normal version of the gene, and it's carried on the X chromosome. So on the X, we put a little superscript for the gene. So this is letting us know that it's a sex-linked trait. His Y doesn't have an H because, remember, the Y chromosome doesn't carry that information. We're just putting it there as a placeholder. But again, there's no H here because there's no information for blood production and red blood cells on the Y chromosome. The mother in this scenario is a carrier, so she has one normal copy and one copy for hemophilia for the disease. So then you just bring these together just like you would in a regular punnett square. And again, using the X's and the Y's. And if you interpret this, this would be a normal daughter because she's got two normal copies. This would be a normal son. He's got the one normal copy, and then the Y. This would be a daughter who's a carrier. She doesn't have the disease because she's got one normal copy. So remember, she gets two chances. She gets one normal copy, one recessive copy. This one normal copy will mean she doesn't have the disease. And then this guy here, this boy, will have the disease because he got the one copy of it that's the disease copy. So again, because the Y has nothing on it for this controlling this condition, he doesn't have any way to mask that. So this child, this boy, would have hemophilia. So only a boy from these two parents would ever have hemophilia. So again, if you're looking at a pedigree, you're going to tend to see more boys because a carrier mother, even if the father is not carrying the disease, will be able to have a son who has the disease.